Oh, thank you. I'll come on stage again. Thanks for the invitation. It's very exciting to be here. Thanks everyone for coming too. So as Thomas mentioned, I'm an evolutionary biologist, and my research is motivated primarily by the observation that no species evolves in isolation, that every organism on Earth is deeply embedded in this web of coevolution with many other species with which it interacts. And these interactions can be mutually beneficial mutualisms all the way towards antagonistic interactions like predator-prey interactions or host-pathogen interactions. And perhaps some of the most widespread and consequential interactions between species in the biosphere are those that have evolved between microorganisms and their eukaryotic hosts. So the Earth formed about 4.5 billion years ago, and within about 1 billion years, the first microbial life, the first life, which was microbial life, arose on Earth. And for billions of years on Earth, all life on the planet was microbial life. And it's only been in the last 500-600 million years where multicellular eukaryotes have evolved. And so the point of this slide is to simply say that all animals, plants, fungi, eukaryotes really grew up in a world dominated by microorganisms. And so it's perhaps not surprising that if we look around today, we see that many eukaryotes have evolved to engage in symbiotic relationships with microbes. And some of the most interesting are those that benefit the host. So here I'm showing a few examples of this. The bobtail squid, which houses in the specialized light organ Vibrio fischeri that helps the squid bioluminesce. Aphids have this specialized organ, the bacteriocyte, that harbors intracellular bacterium Buchnera that synthesizes essential amino acids from the nutrient-poor diets of the aphids. And plants also engage in relationships with bacteria. So here I'm showing some legumes that have these root nodules in which live nitrogen-fixing bacteria. And so my research is focused on two sort of broad questions. First, how is microbial evolution driven by these relationships with hosts? And the other side of the coin, which I think is perhaps even more interesting, is how has the evolution of complex life, multicellular life, eukaryotes, been driven by interactions with microorganisms? And so I think this is a question that should be of interest to all of us because we're all hosts to these complex communities of microbes. So each of us sitting here has about 30 trillion bacteria living in and on the body, most of them living in the gut. So this is showing a picture of human bacteria assembling on a food particle. So if we've eaten lunch today, this is going on within all of us right now. And we've known that these bacteria, the gut microbiota, play important roles in digestion for a long time. But in the last 5, 10, 15 years or so, we've learned that the gut microbiota is deeply integrated with virtually every phenotype that you could imagine. So through these experiments in germ-free organisms, people have shown that the microbiota regulates the development of the circulatory system, so angiogenesis in the intestine. It's deeply integrated with the development and training of the immune system. And there's some work that's come out in the last few years that has shown that the microbiota can actually influence the moods and behaviors of hosts. So every system, tissue and organ system in the body seems to have some relationship with the microbiota. So we know the microbiota is deeply integrated with the host, but we don't really understand the evolutionary history that has produced these relationships. And that's because most of the research that's focused on the microbiota is focused on humans or mice. It's been a largely medical research. So there's really a need for these broad comparative studies in non-model organisms to try to understand how the microbiome has evolved and how these relationships have evolved. And so that's hopefully what I'm going to tell you about today. My talk has 3 parts. So in the first part, I'm going to be telling you about how the community composition of the gut microbiome has changed throughout mammalian evolution. So sort of a descriptive tale of how the microbiome has changed, starting within hominids, humans, and apes, and then zooming out and looking across about 100 million years of mammalian evolution across placental mammals. In the second part of the talk, I'm going to look at some of the processes that are responsible for producing differences in the gut microbiomes of different host species. And in particular, ask this question: do gut bacterial lineages co-diversify and co-speciate with mammals? So in the first 2 parts of the talk, I'm going to be looking at how relationships with hosts have influenced the evolution of microbes and microbial communities. But in the last part of the talk, I want to look at the other side of that coin and ask, how has the gut microbiota influenced the adaptation and evolution of mammals? So I'll tell you about an experiment we've done in germ-free mice relatively recently that I think gets at this question. Okay, so the first part of the talk, how has the community composition of the gut microbiota changed throughout the diversification of mammals? So I'll start with an example from hominids, looking over the last 15 million years or so, and then zoom out across 100 million years of mammalian evolution. This first part is published. This should come out this week, actually, if you're interested in PNAS. And so in the first part of the talk, the basic approach that I'm going to be taking is a comparative approach. And the idea is pretty simple. So if we sample the gut microbiomes of large populations of host species whose phylogenetic relationships are known, we can use the host phylogeny to reconstruct the ancestral state of the microbiome at different nodes in the host phylogeny, and also to reconstruct the changes in the composition of the microbiome that occurred along each branch of the host phylogeny. So it's a simple comparative biology approach. So to do this in hominids, we had to collect first a lot of fecal samples from African apes residing throughout equatorial Africa. So we collected fecal samples from gorillas and chimpanzees living in Cameroon and Gabon, bonobos living in the Democratic Republic of the Congo, also eastern gorillas living in the DRC, and then a population of chimpanzees living in Gombe, Tanzania. And from here, we use the standard 16S profiling approach, which I'm sure many of you are familiar with, where you take your microbiome sample. Here it's a fecal sample. You amplify the 16S rRNA gene, a universal gene present in all bacteria, sequence it on an Illumina machine, and at the end of the day, you get a sense of all the bacterial taxa that are present in that sample and the relative abundances. So here I'm showing two hypothetical examples of two different samples. So these aren't real data, but here each bar represents a different sample, hypothetically, and each color would represent the different proportion of the proportion of a different bacterial taxon. Okay, so in this slide, I'm going to show many of these stacked bar charts. And so this one slide is my entire PhD in one slide. If you want to fall asleep after this, you're good to do so. So here are what the data look like for gorillas. So here, each bar represents a different gorilla, and each color represents the relative abundance of a different bacterial class. So it's a very high-level taxonomic view. But you can see gorillas have this immense amount of bacterial diversity, this technicolor pattern. Here are the data from bonobos, from chimpanzees, and then we combined our data from African apes with previously published data sets using the same methods from humans produced by Jeff Gordon's group in Washington University in St. Louis. So humans living in the Amazon rainforest in Venezuela, living very traditional lifestyles. Humans living in a rural community in Malawi, and then also humans living in urban centers in the United States. So we were able to compare our data with humans representing a range of populations and lifestyles. So if you sort of blur your eyes a little bit and just look at this, you can see right away that each host species harbors a distinct gut microbiome composition from these data. So gorillas have a ton of diversity here, and then if you look all the way on the right, humans living in urban centers in the United States are essentially a swath of this red. Another way to visualize the data is through principal coordinates plots. So this is showing PC1 and 2 of the Bray-Curtis distances among all the samples. And again, the point of this slide is just to show that each host species harbors a distinct microbiota. So chimpanzees and gorillas are clustering up here. Gorillas are clustering here and actually come out on the third PC axis away from the board. And then humans from Malawi and Venezuela are clustering here. And then this separate cluster of humans is humans living in urban centers in the United States. And we think that these differences among the gut microbiomes of host species aren't just due to environmental effects. And part of the reason we think that is because when we look at populations of chimpanzees and gorillas living in sympatry, so living in the same geographic location, nesting in the same trees, eating many of the same foods, we see that they still maintain compositionally distinct gut microbiotas from gorillas. So here the circles represent chimpanzees, the squares represent gorillas, and the circles in blue represent chimpanzees who live sympatrically with the gorillas. So we see there's a subtle convergence between sympatric chimpanzees and gorillas, but the vast majority of the differences are maintained in sympatry. So using these data, we can begin to ask how has the composition of the microbiome changed throughout hominid diversification. So here I'm showing on the y-axis a measure of microbiome dissimilarity, the Bray-Curtis dissimilarity between hosts. And on the x-axis, I'm showing the divergence time between hosts. So if we look within species, gut microbiomes tend to be very similar. If you compare bonobos with chimpanzees, they tend to be slightly more dissimilar. And if you compare bonobos and gorillas, they're very much dissimilar relative to the other two comparisons. And it looks like, at least in these species, there's been a relatively clock-like rate of microbiome divergence over the course of African ape diversification. So we can also plot humans onto this line and ask, how has the microbiome evolved or changed in the human lineage. And when we do that, we see a slightly different pattern. So humans living in urban centers in the United States harbor gut microbiomes that are more divergent from the ancestral state than we would expect based on our comparisons to African apes. So this is comparing humans in the US versus chimpanzees. You see they're more divergent than we would expect based on the divergence times of the hosts. And humans in the United States versus gorillas, they're more divergent than we would expect based on the divergence time separating the hosts. So there seems to have been a rapid transformation of the gut microbiome in humans, at least in the United States. So this rapid change in human gut microbiomes, it could be due to the increase in the abundance of some taxa or the decrease in abundance of other taxa. So we wanted to actually dig into the details of what's actually underlying this rapid divergence. And so again, to do this, we're going to use a phylogenetic approach, a comparative approach. And the basic idea here is that for each bacterial taxon, we can look at the distribution of, or the abundance of that taxon in each host species. So if we were to see a pattern like this, where the abundance of this taxon was very low in humans, but very high in the African apes, we could infer that the relative abundance of the taxon was high in the ancestor of humans and the African apes, and that there's been a reduction or a decrease in the abundance of that taxon in the human lineage. And we can do that for every bacterial taxon in the gut, and also we can do it for every branch of the host phylogeny, and not just look at things that decreased, but also things that increased as well. So it's essentially a parsimony reconstruction of the changes that have happened in humans. And so when we do that, we find many bacterial genera that distinguish the different host species. And so I'll just highlight a few of the ones that I think are the most interesting. So if we look along the human lineage, we see there's been an over 5-fold reduction in Methanobrevibacter, an over 5-fold reduction in Fibrobacter, and a corresponding over 5-fold increase in the relative abundance of Bacteroides within humans. So these types of changes are consistent with the known dietary transitions that occurred during human evolution. So Methanobrevibacter and Fibrobacter, which have been reduced in humans, are known to play a role in the digestion of plant polysaccharides, whereas Bacteroides within human populations has been associated with meat eating. So this is consistent with the idea that human microbiomes sort of been specialized for a meat-based diet. And I should add too that these changes that distinguish humans, they distinguish all the human populations that we looked at. So it's not something just specific to the United States. All human populations have these gut microbiome features relative to African apes. Another pattern that popped out immediately from the data was that there's been a reduction in microbial diversity within the human gut microbiome since humans and chimpanzees diverged about 6 million years ago. So this is just showing the number of bacterial genera per individual in wild apes, and then the number of genera per individual in humans from Malawi, Venezuela, and the United States. You can see that humans always harbor less bacterial diversity than apes, and that humans in the United States in particular harbor the lowest levels of bacterial diversity of any human population. This isn't just true at the genus level, it's true at every taxonomic level. So here again, I'm showing the diversity in apes and in humans in red, at the phylum, class, order, family, genus, and species level. And in each case, humans have less diversity than apes. And moreover, humans from the United States have the lowest diversity of all. So this is all consistent with this idea that there's been a loss of microbial diversity in humans since humans diverged from apes. So some of the outstanding questions from these results are why? What is causing the loss of microbial diversity from humans? So I can talk more about that later if people are interested, but we have some good reasons to think that both antibiotic use and the lack of dietary fiber might be playing a role. But perhaps more interesting is what are the consequences of this loss of diversity? And one of the hypotheses that seems to be gaining more support is that this loss of diversity may actually be bad for us, at least in urban populations. So this hygiene hypothesis, where it's articulated nicely by Marty Blazer in this book, the basic idea is that because we've lost some of these microbes that we've evolved with for so long, that we've actually predisposed ourselves to a lot of modern diseases like autoimmune diseases and metabolic diseases like obesity. So I think this is an exciting area for future studies. Okay, so in this very first part of the talk, I showed you that in African apes, the compositional change in the gut microbiome over evolutionary time seems to have been fairly steady, but that humans have experienced a rapid transformation of the gut microbiome, and that's marked by a reduction of bacterial diversity, a loss of bacterial diversity. So I want to take the same kind of approach and apply it much more broadly, looking across all placental mammals, or a lot of placental mammals, over about 100 million years of mammalian evolution. And because we're sampling many more species here, so we've collected fecal samples from 17 different species, from populations of 17 different species residing throughout North and South America. So we have wolves, coyotes, bears, mountain lions, bobcats, deer, moose, elk, caribou, and then a range of rodent species. And I was going to say, because we've sampled so many more species here, and we're not just relying on comparisons with a few species, we can actually start to get at this question of what is driving the divergence between the gut microbiotas of mammalian species. So we're going to be comparing the microbiotas of different host species to ask how the microbiome's changed over evolutionary time. So I'll show you all the data at once to begin with. So here I'm showing on the y-axis the Bray-Curtis similarity, or a measure of compositional overlap between the microbiotas of host species. Each dot represents a comparison between two host species. And then on the x-axis, I'm showing the divergence time. So we see a similar pattern across all of mammals that we saw within hominids. As divergence time between host species increases, the microbiotas become more dissimilar, or the overlap decreases. But you can see there's a lot of variation around this exponential decay curve. So we would like to be able to explain some of that variation. And it turns out that whether or not the host species live sympatrically or allopatrically seems to determine in part the rate at which their gut microbiomes diverge. So when we look at just allopatric pairs of host species, so species living in two different geographic regions, we see the rate of microbiome divergence is much faster than the rate of microbiome divergence in sympatric species, shown here in dark gray. So moreover, within mammalian communities, we can actually dissect this even further and show that species that live in the same mammalian community in the same region that are connected by a predator-prey relationships actually harbor more similar gut microbiotas than you'd expect based on the phylogenetic divergence between the hosts. So it seems that contact between host species is actually slowing the rate at which their gut microbiomes diverge even further. So I want to dive into that result a little bit more because I think it's interesting. So we were able to compare predator species with different prey species on which these predator species either feed or do not feed. So here, while also controlling for the host phylogeny, or the predator phylogeny. So here we have mountain lion and bobcat, and wolf and coyote. So mountain lions and wolves rely heavily on these large-bodied artiodactyls, like Cervicolaphus, but the bobcats and coyotes typically don't feed on these large-bodied artiodactyls. And we see that reflected in the gut microbiome dissimilarity. So here's Bray-Curtis dissimilarity on the y-axis. Mountain lions and wolves, the microbiotas are more similar to Cervicolaphus than are the microbiotas of bobcats and coyotes. And we see that for every comparison in our data set with the large-bodied artiodactyls. And the converse is also true. If we look at small-bodied predators and small-bodied prey items, we see that the small-bodied carnivores always harbor more similar gut microbiotas with the small-bodied prey than do the large-bodied carnivores. And we have some reasons that I can also talk about at the end if there's interest, that we don't think this is due to body size effects alone. So we can compare the carnivores to large-bodied and small-bodied prey species that live allopatrically, and we don't see the same pattern. So this really suggests that predator-prey relationships are providing routes of bacterial transmission between very distantly related mammals, and that this transmission is actually slowing the rate at which the microbiotas diverge over evolutionary time. Okay, so hopefully I've convinced you at this point that the microbiotas of host species diverge compositionally over time. And at least in mammals, it looks like the geographic or spatial separation between host species accelerates microbiota divergence. Living sympatrically slows it down a little bit relative to allopatrically, and predator-prey interactions enable the transmission of bacteria between host species and can slow microbiome divergence over evolutionary time as well. Okay, so far I've shown that there seems to be very strong relationships between host phylogenetic history and the composition of the microbiome. But I think it's important to point out that multiple processes can produce these associations between the composition of the microbiome and the host phylogenetic history. And so here I'm showing just two possible processes that can produce these associations. So on the left is the process of co-diversification. So in this case, these four species share common ancestors at some point in the past. Back here, this common ancestor had a microbiome with these bacteria. As this ancestor diverged into different lineages, the microbes were vertically inherited and diversified alongside the host lineages, leading to a pattern in the present day where more closely related host species harbor more similar gut microbiomes. So that's one process that can produce these associations. Another non-mutually exclusive process is shown here on the right. So we know that more closely related host species tend to be more similar in all sorts of traits. Gut morphology, immune system, diet, et cetera. So it could be that more closely related host species are actually selecting more similar sets of microbes from some shared environmental pool. And that can also produce this pattern where more closely related host species have more similar gut microbiomes. So this pattern doesn't require any long-term associations between the microbes and the hosts. In this case, the fitness of the microbes is totally disconnected, for the most part, from the fitness of the host. The microbes can live just fine in the environment, whereas here, the fitness of the microbes and the host are very much in line with one another. So I'm very curious to try to differentiate between these two processes to see which one's driving the patterns that we see. So that brings us to the second part of the talk. We're going to ask, do gut bacterial lineages co-diversify with mammalian species, looking in particular in hominids? And so before we did this work, most of the examples of co-diversification between bacteria and animals came from insects. So things like aphids, where bacteria are vertically inherited, and they're very much determined by maternal inheritance and host genetics, and they're really not labile at all. In contrast, the mammalian gut microbiome, the human gut microbiome in particular, we know that it's very influenced by external factors. So diet, how much sleep you get, what medications you're taking, the list goes on and on and on. So it wasn't clear when we started whether bacterial lineages could persist over evolutionary timescales required to produce co-diversification. And so what we're going to be looking for to see if this is happening is for congruent phylogenies. So this is really the hallmark of an ancient and intimate association between host lineages. So we know the phylogeny of humans and African apes. We're more closely related to chimps, and then gorillas are the next group out. And so we're going to be looking in the gut microbiome and asking, do bacterial phylogenies mirror the host phylogeny when we look across these different host species? But we ran into an issue when we tried to do this initially with 16S data, and that's that 16S amplicon sequencing doesn't provide enough resolution to build bacterial phylogenies over the timescale of hominid evolution. And that's because they just evolved much too slowly. So sequence divergence in 16S accumulates at something like 1% per 50 million years. So hominid evolution is only about 15 million years old. So this doesn't provide enough phylogenetic signal. So we had to turn to a different marker gene in order to answer this question. And so we came up with this gene gyrase B, which is a protein coding gene. It evolves much more rapidly than 16S because it has silent sites like protein coding genes do that are more labile evolutionarily. So the basic approach is just like the approach for 16S sequencing I outlined previously, but here instead of targeting 16S, we're targeting this gyrase B gene. And at the end of the day, instead of getting a list of the different taxa and their abundances, we're going to get a catalog of bacterial strains and the relative abundances of those strains within the samples. So we applied this approach to sequence 3 bacterial families residing in humans, gorillas, bonobos, and chimpanzees. So we targeted the Bacteroideaceae, the Bifidobacteriaceae, and the Lachnospiraceae. And together these make up about a third of the human gut microbiome. So I'm just going to jump right into the results for the sake of time. So first I'll tell you about the Bacteroideaceae results. So we built bacterial phylogenies for all the sequences we recovered. And we recovered 3 clades of Bacteroideaceae from our data set. And so here, each branch represents a different bacterial strain, and the branches are colored based on the host species from which they were recovered. And we see that in each case, in each of these clades, the bacterial phylogeny mirrors the host phylogeny. So here, gorillas are the outgroup to a clade containing chimpanzees and bonobos. It appears the human representative of this clade has been lost, at least in the humans we sampled. Same for the second Bacteroideaceae species. And then here, Bacteroides vulgatus also shows a congruent phylogeny with the host. So we also looked at the Bifidobacteriaceae and found the same pattern. So the phylogeny of the bacteria matches the host phylogeny much more than you'd expect by chance. In this case, it's almost perfect with one exception. There appears to have been a transfer from bonobos into gorillas of this Bifidobacteriaceae. But altogether, these results indicate that multiple Bacteroideaceae and Bifido lineages have co-diversified in parallel with humans and the African apes. So it shows these associations are ancient. They predate the diversification of host species, and that hosts have maintained specific associations with different bacterial lineages as they diversified. So we also looked at the Lachnosporiaceae. It was the last group we looked at. And in contrast to the previous two families, this family contains a lot of spore-forming bacteria. So you can see this here if you know what to look for. So they form these hard spores that protect the bacteria from harsh environmental conditions found outside the gut. And when we look at the phylogeny of the Lachnosporiaceae, we see absolutely no evidence for co-diversification whatsoever. So this is all the colors are jumbled up. This phylogeny does not match the host phylogeny more than you'd expect by chance. And so it shows that Lachnosporiaceae lineages appear to move across host species boundaries fairly regularly. So together with the previous results, this shows that the gut microbiota is really a composite of two different types of lineages. One type that's co-diversifying and maintained within host lineages, and then the other type of bacteria that is independently diversifying and can be transferred across host species boundaries. And I think these phylogenetic approaches hold a lot of promise for dissecting the microbiome into these two components. Okay, so to answer the second question, yes, it looks like gut bacteria have in fact co-diversified with hominids. It's possible this is going on across many other mammalian species, so we're looking at that now. I think it'll be interesting in the future. And I only showed results from 3 bacterial families, but there are many more. There are potentially dozens or even hundreds of lineages co-diversifying in parallel, so we're just beginning to scratch the surface of this. Okay, so in the first 2 parts of the talk, I was focused mainly on how associations with hosts have influenced the evolution of bacterial communities and also bacterial lineages. But in this very last part, I want to try to address this question: how is the microbiota influencing mammalian phenotypes, fitness, and ultimately adaptation? So does the microbiota actually act as a selective agent or selective force in mammalian evolution? Have mammals adapted to their gut microbiotas? And to do this, I've just started working with germ-free mice. I think they're a really great system for trying to get at some of these questions. And we're going to be doing microbiota-transplant experiments in germ-free mice. So here I'm showing a couple of isolators. In these isolators are cages of mice that are devoid of bacteria. And I want to ask, does the gut microbiota drive adaptation in these mouse species? And to do that, we're going to rely on 3 species of mice. So we have in the lab now 3 different species of mice: Musculus domesticus, the common house mice, which is also the common lab mouse. We have its sister group, or sister species, Mus spreadus, and then a more divergent. So these are about 1.5, 2 million years diverged, and then a more divergent species, Mus bahari, which is about 7 million years diverged. And we found in the lab, in the same room, eating the same diet, that the different species harbor compositionally distinct gut microbial communities that reflect the host phylogenetic history. So within species, this is again microbiota dissimilarity on the y-axis. We see the microbiotas are very similar. If we compare the sister species, Spreadus and Domesticus, we see more divergence, but not quite as much divergence as we see when we compare the microbiotas of this clade with Mus bahari. So this is mirroring the pattern that we saw across all the other mammals, where host phylogenetic divergence scales with microbiome divergence. And so the experiment that we want to do is to take the microbiomes of these different host species and to transplant them into a germ-free Mus musculus domesticus background, and to ask how these different microbiotas affect the fitness of germ-free Mus musculus domesticus. And the prediction is, if the house mouse has adapted to its gut microbiome since diverging from Mus bahari 7 million years ago, that we should see the house mice should have the highest fitness when we give it its own Mus domesticus microbiota. Its fitness should be slightly reduced by the Mus spreadus microbiota, and its fitness should be reduced even further by the Mus bahari microbiota. And here we're going to be measuring fitness simply as growth rate. So growth rate in mice is a pretty good indicator of fitness. Larger mice tend to be more fecund, to have higher fitness in the wild, so that's going to be sort of our proxy for fitness. So we did this experiment, and we found that each of the 3 donors, microbiotas, they colonize domesticus equally well, at least in our experiments. So here I'm showing the 3 different donors we had. This is the domesticus donor, the spreadus donor, and the bahari donor in the circles. And then in the triangles, I'm showing the microbiotas of the recipients. So you can see that in each case, the recipient's microbiota very closely mimics the microbiota of the donor, the spreadus and the bahari. And moreover, that this pattern of phylosymbiosis or congruence between the host phylogeny and the microbiome composition, it's maintained in these germ-free Mus domesticus recipients, or gnotobiotic recipients. So the domesticus treatment is more similar to the spreadus treatment than it is to the bahari treatment. So we're able to see pretty good reconstruction of these microbiomes in germ-free Mus domesticus. And it's interesting, I think, that each microbiome colonized equally well. So this is a measure of colonization success, simply the percentage of bacteria that colonize the host, the percentage of bacteria in the donor that end up in the recipient. We see there's no significant difference among our treatments. We don't see any significant differences in bacterial load among our treatments or bacterial diversity. So as far as we can tell, at least with the power that we have, there seems to be no real difference among the different microbiotas colonizing the host. So we get good colonization in each case. But where we do see an effect is when we look at the growth rate of Musculus domesticus. So this is showing the fold change in body weight on the y-axis against age from 4 weeks, which is a couple of weeks after we did the transplant, to 10 weeks of life. The Mus domesticus that received the Mus domesticus treatment grow very well, just like you'd expect. The Mus domesticus that received the Mus spreadus treatment grows slightly and significantly slower than the Mus domesticus treatment. And then the Mus bahari treatment grows the worst. And actually, I haven't included in this graph, but we just got the data back from the negative control. These Mus bahari treated Mus domesticus mice are actually growing slower than germ-free Mus domesticus. So it's actually worse to have a bahari microbiome than no microbiome at all in our experiment. And this is consistent with our hypothesis that Musculus domesticus has adapted to its gut microbiome over the last 7 million years or so. Okay, so with that, I just want to summarize the main points of the talk. The gut microbiotas of host species diverge compositionally over evolutionary time. And I think we can say with a lot of confidence now that is a general pattern that we see across all mammals. And it's possibly true across all vertebrates. But we see that the rate of microbiome divergence between host species isn't fixed. It varies depending on sort of the ecological scenario in which 2 host species are diverging. So geographic distance between host species accelerates microbiome divergence, and predator-prey interactions can slow microbiome divergence. In hominids, there are gut bacteria that co-diversify with hominid species. And this process of co-diversification is, I think, part of what's producing this divergence of the microbiota between host species. And co-diversification, again, it could be happening across mammals or across vertebrates. This is something we're interested in investigating now. And lastly, these results from these experiments in germ-free mice suggest that mammals are actually adapting to their microbiotas over evolutionary time. So in house mice, it appears house mice have adapted to their gut microbiota as Mus species diversified. So in the future, I'm really excited about doing more of these experiments to try to understand how the gut microbiome has played a role in the adaptation of mammals. So I'm really excited to talk to more of you as I'm here, and I hopefully can have some good questions now. But before that, I'd like to acknowledge collaborators. So these are a lot of people who helped along the way with the different experiments. My thesis committee, other collaborators, and my PhD advisor and my current postdoc advisor, and then a lot of funding sources. So with that, thank you.